In today's lecture, we will learn some very important genetic terminology, then how to make monohybrid crosses, how to make dihybrid crosses, and what are the laws of Mendelian inheritance. Yes, this information is not everyday information and you might not have heard about these things before, but as we go through this lecture, you'll understand how easily you can interpret what characters will be inherited by the children if you know what are the characters of their parents. Since we are studying Mendelian inheritance, so it's important to know who was Mendel. Mendel, or Gregor Mendel, was an Austrian monk. He is known as the father of genetics. This monk had a pea garden. He would roam around this garden and see that pea plants have very distinct characters. So he made a list of those characters. Those characters were related to the shape of seed. The seed would either be round or wrinkled. Then the color of seed, it would be either yellow or green. Then the flowers of pea plant, they would either be white or violet or purple. Then he can see a very clear contrasting character of pod. The pod would either be full like this or constricted like this. And he could see a distinction between the color of these pods. The color would either be yellow or green. Then he could see very contrasting characters related to stem. The stem would have either the flowers present axially or terminally. And related to the size, he could see that either they were long or short. So he could see distinct contrasting characters present in the pea plant. Then he chose to study these plants. Why did he choose pea plants? One, because he could see very clear contrasting characters which were easy to record. And then because he had this garden already, he could control the self-pollination, cross-pollination, if he wanted to make the two plants breed with each other or crossbreed, he could control all those conditions. And another very important point is that the pea plant had shorter lifespan and they could produce their offsprings or their next generation or seeds in a short amount of time. So what are broadly the concepts behind the genetics? Every organism, here we are talking about a plant, is made up of tissues. Tissues in turn are made up of cells. Plant cells are present in plants. The plant cells have outer cell wall, which you are seeing here, and a cell membrane, the inner one. They have, like all other living cells except the exception of few, for example a red blood cell, almost all living cells have nucleus. So the nucleus is the main organelle that contains genetic material in the form of DNA. So in nucleus there are chromosomes. Chromosomes are the condensed versions of DNA. So a very long strand of DNA is wound around some proteins and they condense to form chromosomes. So there are proteins and a long strand of DNA is wound around them to form chromatin, and chromatin condenses to form chromosomes. Now let's try to understand what exactly we are talking about. There are chromosomes, and let's say this chromosome came from your father and the other chromosome came from your mother. So in human beings there are 46 chromosomes and they exist in 23 pairs. So these 23 pairs of chromosomes have one coming from father and one coming from mother. They are called homologous chromosomes. Why? Because they not only have same shape, they can be like this or like this or like this. But a homologous pair would always have same shape, size, and also similar genes. So genes are fragments of DNA. Fragments of DNA. And let's say this small fragment of DNA is a gene. Then this small fragment of DNA is also a gene. But this came from your father and this came from your mother. Now let's say we are talking about a gene related to hair color. So this gene will also have information about hair color, and this gene will also have information related to hair color. Let's say this fragment is related to eye color, and this is also related to eye color. So these genes are related to eye color. Now, one gene came from your father and one gene came from your mother. If we talk about hair color, let's say father had black hair and mother had brown hair. So you have two options. You can have either black hair or brown hair. So gene is a trait, and the options that you have are alleles. The alleles are represented as B, B, or any other letter. You can pick any letter to represent any gene. Let's say you're talking about eye color, then you can say I'm going to call the gene that came from father is capital E, and the one that came from mother is small e. But how do you decide if the one that came from father would be a capital E and the one that came from mother would be a small e? There is no rule like that. What the rule says is the dominant allele will be represented by capital and recessive allele will be represented by small letter. Now, first you need to know which character is dominant. Is the black hair color of father dominant or the brown hair color of mother dominant? If the black hair of father is dominant, then black will be capital B and brown will be small b. If brown color is dominant, then you will represent brown as capital B and black as small b. But you don't necessarily have to use B. You can say, okay, I'm going to say that the brown color is represented by W and black color, which is recessive, is represented by small w. So always remember that the dominant character will be represented by a capital letter. It can be any letter, and the recessive character is represented by small letter, which is A. The other thing is, now one gene has come from your father and one has come from your mother. The combination present in you can be one dominant, one recessive, or both dominant. Maybe both of your parents had the same allele, or the both can be recessive. Maybe both of your parents have recessive allele in their genome for hair color. So what will be the options? If your father has, if we have already decided that black is dominant over brown, and you're going to represent black as capital B and small b represents brown, then maybe the character that came from your father is dominant and mother is recessive. Or maybe the one that came from mother is dominant and father is recessive. Or both of them had dominant allele, or both of them had recessive allele. So this condition is called heterozygous, and this one is homozygous, and this one is also homozygous because they have same alleles, and here they are different, so they are hetero. But this is homozygous dominant and this is homozygous recessive. All right, so we'll study about all these terminologies in the subsequent slides. All right, so Mendel started his experiment with the pea plant. Let's say he was observing one of the characters, which is the color of pod. The color of pod he found was either green or yellow. So he self-pollinated these plants, did not let them cross with these guys, and after only self-pollination, he found that all the generations would only have green pods and this side had all the yellow pods. So he called them pure green pod line or pure yellow pod line because they came from green parent or yellow parent and they all were either green only or yellow only. They did not have any mixed characters. So this is how he established his parental line. Now these are the parents which are pure. Pure means if we try to guess, maybe green color is both capital G, and this one is both small g, small g. But till now, Mendel did not have the concept of dominant or recessive or how to put these letters G, capital G, small g. And when we talk about these capital G, small g, what are these called? These are called genotype. That means we are trying to guess what would be the allele coming from their father and mother. And when we know that the final color here is green and final color that you see here is yellow, so green and yellow are phenotypes. So the character that you see is the phenotype, and the letters that you are trying to guess on the basis of which you say, okay, this is the color that I see, this must be the genotype. So the letters that you're guessing ends up making the genotype. Okay, then he crossed these two parental lines. Now he's making the green pod color plant and yellow pod color plant to cross pollinate. He saw that after cross pollination, it was not like he would get some yellow, some green, some yellow, some green plants, or a mixture like a yellowish green plant, so that there's a blend of two characters and then you get an intermediate color, so mixing yellow and green. So if you take yellow and green color in a palette, you would see mixing these two colors give you a yellowish green color. So that was also not what happened in the crosses from these two plants. What he actually saw is all of them were green. Then what happened to the yellow color? Where did that go? Even though one parent was green and other parent was yellow, all the progeny, all the generation was green only. So he called green color as dominant and yellow color as recessive. So he said that one color or one character is dominant over the other character, and that's how he established the law of dominance, which says that the dominant character will overshadow the recessive character, and that's how you're able to decide which character is dominant. You take the pure parental line, cross them, and the generation that comes out of it, if it's all green, then green is dominant. If it's all yellow, then yellow is dominant. So this is his first generation or first filial generation, or also called the F1 generation. Okay, then he took this F1 generation, which actually came from a cross between green and yellow parent, and this also came from a cross between green and yellow parent. So these are not pure lines, they have come from the cross between a green and yellow, whereas the yellow pure line or a green pure line comes from only green or only yellow parents. All right, so when he crossed these two, suddenly he found yellow is back. So he got majority green plants, but in minority he also got these yellow plants. And that's surprising. Although the parents were green colored only, the progeny could still have a yellow color plant. So that means the yellow color, which was hiding, has not completely lost its identity or wasn't like completely gone. That information was still there, but it was just hiding. So in the F2 generation or the second filial generation, yellow color or the recessive color is back again. So Mendel said that the factors do not get mixed up, but they appear in later stages. So the yellow color did not lose its identity, it appeared in F2 generation, and this established the law of segregation, that the characters are actually able to separate from each other. They are not like sticking to each other or not getting blended up or mixed up with each other. Now let's see what is happening at the gene level. So pea plant has 7 chromosomes, means 7 pairs of chromosomes. So 1, 2, 3, 4, 5, 6, 7. And let's say on one of these chromosomes, which is this one, you have the gene for pod color representing green pod color. And the mother plant, this is the father plant and this is the mother plant. Mother plant also has 7 chromosomes, and on the 6th chromosome you have the allele for yellow pod color. So if you zoom in, this is a homologous pair of chromosome in this plant. This is a homologous pair of chromosome in this plant. That means this plant also received one from its father and one from its mother, and this also received one from its father and one from its mother. So it's a generation to generation thing. We also have to think like backwards. Where did they came from? So this is a homologous pair of chromosomes. At specific location they have same gene. Here we are talking about the gene related to the pod color. So these are 2 alleles, 1 allele, 2 allele. one allele is capital G, the other allele is also capital G. This is the pair of chromosome in the mother plant, and one came from her mother and one came from her father, and this is a homologous pair of chromosomes. So at one specific location they have the gene for the pod color. Here they have received both the alleles to be recessive ones, and both are small g. That's why the color presented here is yellow. If they had both capital G's, then the color presented would be green. So now you understand what is a gene. So this is a gene, and what is the allele? Allele is the options that you have. You can have G or G. So these are the two alleles of one gene related to the pod color. So according to these options, if you have GG, you have green pod color, and if you have GG, then you have yellow pod color. Okay, now this father undergoes meiosis, and this mother also undergoes meiosis. They produce gametes here, and this mother also produces gametes here. For example, the father would have produced pollen and mother would produce the ovule. So in the pollen you would have half the number of chromosomes. Half goes here, half goes here. So the pairs that you had in the beginning have now separated into half and half. And here also the chromosome pair that you had in the beginning have now separated into half and half. So the pure parental generation produces one kind of gamete. That means the capital G and this capital G are now separated. This is gone in one of the gametes and this has gone in one of the gametes. And here also the two alleles have separated. One is gone in one gamete and the other is gone in one gamete. So these have separated from each other. They are not influencing, they are not like sticking together. Both the G's are not going in the same gamete, or both the capital G's are not going in the same gametes. They have separated from each other. So now let's look at the Mendelian monohybrid cross. So monohybrid cross helps us in understanding how the characters from parents can help you decide what will be the characters in the children. So let's start with the parental generation. The parent here, as we just studied, had both capital G's. This is called the homozygous green parent, and this one is the homozygous because both the alleles are same, so they are homozygous. If one was capital G and one was small g, then it will be called heterozygous. So here homozygous yellow parent. When they produce their gametes, one gamete will have one kind of allele, other gamete will have other kind of allele. Here also the alleles get separated into two different gametes. Now for when you have to produce the first generation, what happens here? There will be fertilization. That means the gamete of father will have to fuse with the gamete of mother. So either this gamete of father fuses with this gamete of mother and you get this combination, or this gamete of father fuses with this gamete of mother and you get this combination, or this gamete of father fertilizes with this gamete of mother and you get this combination, or this gamete of father fertilizes this gamete of mother and you get this combination. So in every combination, a capital G is combining with small g, capital G combining with small g, capital G combining with small g, capital G combining with small g. And we know that capital G representing green color is dominant over small g representing yellow color. So the color that you finally see is the green color. Here also green, green. All of these pods will be green because green is dominant over yellow. So all the generation, F1 generation, the genotype of all these plants is capital G, small g, heterozygous, but all of them 100% heterozygous. And what is the phenotype? Phenotype is green color. All of these have green color. So we have established that green color is dominant over yellow color. Again, remember the combination of letters, or what are the letters in the genes of an organism, gives you the genotype. And what do you see in the physical form? The final character that is the result of these letters is the phenotype. That means the final result of these characters, which will be either green color or yellow color, that is the phenotype. Alright, now we had this F1 generation in which you had heterozygous characters, which means all of them had one capital G and one small g. That's what we just saw, right? And all of them were green in color. The phenotype was green. Now if we make a cross between these, we are making this plant fertilize with this plant. But before fertilization, they have to undergo meiosis to form the gametes. So the gametes will have half the number of chromosomes. This chromosome, this homologous pair of chromosome, gets separated in the two gametes. So one will be capital G and other will be small g. One will be capital G, one gamete, and the other gamete will have a small g. Now let's make a cross between these. If this one fertilizes with this one, you get capital G, capital G. If this one fertilizes with, if this one fertilizes with this one, you get capital G, small g. If this one fertilizes with this one, you get capital G, small g. And if this one fertilizes with this one, you get small g, small g. Now let's see what is the genotypic and phenotypic ratios. You got one plant this type, two plants this type, and then one plant this type. So these are the genotypic ratios. But let's see the phenotype. Although you have two kinds of alleles here, but the result is green. Here also the result is green. Why? Because capital G is dominant over small g. Capital G is dominant over small g. So what happens is, here also you have green color, green color, green color. So genotypic ratio will be different from the phenotypic ratio. In phenotypic ratio, you would have 3 greens and only 1 yellow. Now let's look at how to make a Punnett square. So the genes or the gametes that we saw in the previous generation, now we have put those gametes on top of a square. Here, 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 and here. So these are, let's go back and see what we are doing. So the gametes that we formed here, one set came from father and one set came from mother. Now we have put them on a square. This set comes from father and this set comes from mother. We are making this cross because it's easier to visualize or easier to make the cross between the gametes of parents by doing this square method. So this one fertilizes with this one, or this one fertilizes with this one, or this one fertilizes with this one, or this one fertilizes with this one. Makes it easier to see, right? So the end results are, you get one capital G, two capital G small g's, and one small g. So the genotypic ratio, in which you're only looking at capital and small letters, is 1:2:1. This condition is called homozygous because they are same. This is also called homozygous because they are same, but this is homozygous dominant and this is homozygous recessive, and this one that had different alleles is called heterozygous. If you look at their phenotype, then what is the result from these letters? This one will be green, this one will be yellow. That's easier to interpret because they don't have any other option. They can only be green here and yellow here. They don't have any other option. But here they have an option. It can be either green or yellow. But as per the law of dominance, the green will be dominant over yellow. So the end result would be green. So green is overshadowing yellow, and you will get, this will be green, these two will be green, so 3 greens and then 1 yellow. So the phenotypic ratio becomes 75% green and 25% yellow. So we have established that green allele is completely dominant over yellow allele, and yellow expresses in homozygous condition only when there is no capital G, then you will get a yellow color because there is nothing there to overshadow this character. So what did we learn in Mendelian monohybrid cross? It's called a Mendelian cross because it follows Mendelian rule of dominance and segregation of characters. It's a monohybrid cross because we are only looking at one character at a time. And it's a hybrid because you had two forms of alleles. You were dealing with G and G. But now we will study a dihybrid cross, in which we will study two genes or two characters or two traits at once. So here we are looking at two different characters. We have picked to study the flower color and the pod color. Why did we pick these two characters? Because we have, if we were studying this, then it'll also be yellow and green and yellow and green. So it's hard to understand which character we are talking about. But here we have white and purple contrast and yellow and green contrast. So if we say white or purple, you know that we are talking about the flower color. And if we say yellow and green, you know that we are talking about the pod color. Now we'll go through the same kind of cross, but for two characters at a time. So follow what I'm saying. Here is a homologous pair of chromosome in the father, and here is a homologous pair of chromosome in mother. At one location there is the gene related to the pod color, and at other location there is a gene related to the flower color. Here also at one location there is gene related to pod color, and at other location there is gene related to the flower color. Here you have both capital G, capital G. Here you have both PP representing purple color, and here you have small p, and small g representing white color of flower and yellow color of the pod. This is a homozygous parent with both the dominant alleles, and this is a homozygous parent with both the recessive alleles. Now, here they have meiosis going on and leads to the formation of two gametes. So this is gamete 1, this is gamete 2, and this is mother's gamete 1, and this is mother's gamete 2. When they cross with each other, so one of these gametes will fuse with one of these gametes to make the offspring. So if this parent fuses with this parental gamete, you get this, and if this gamete fuses with this gamete, you get this. If this gamete fuses with this, you get this. If this one fuses with this one, you get this. Now let's look at the end result. In end result, you have a pair of chromosomes that have pod color gene and the flower color gene. You have an option of green or yellow. You have an option of purple and white. So you have the option for purple or white and green and yellow. 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 So all of them have heterozygous situation. So you have different allele here, different alleles here, different alleles here, different alleles here. So they are heterozygous, but all of them are same. This is same as this, this is same as this is same as this is same as this. So 100% same genotype, and the type is heterozygous. What is the phenotype? Now you know that purple color is dominant over white color. So wherever you have a pair of purple and white, purple will be the end result. The flower that you get on a pea plant will be purple. Wherever you have a combination of green and yellow, you know that green is dominant over yellow, so the pod color will be green because green is dominant over yellow. So out of capital P and small p, you will get purple. Out of capital G and small g, you will get green. So all the plants will have the same phenotype. That will be purple color flowers and green color pod. So here we have followed Mendel's rule, which is that green color will be completely dominant over yellow color and purple color will be completely dominant over white color, and the phenotype that you get will be purple color flowers and green color pod. Now this generation, that was the filial generation that we got in the last cross. Here is the generation that we got in the last cross. In one of the chromosomes it has capital P, capital G, and on the other chromosome it has small g and small p. But it can also be other way around. We are just representing them on the different chromosome pair. Now they are going to produce gametes, because during the meiosis we know that there is crossing over step, which leads to the exchange of a small fragment of chromosome. Let's say there is no crossing over. If there is no crossing over, then you get this type, and this type separated. But if a small fragment of this crosses, this goes here and this goes here, then the result would be green. The white comes with green, or purple goes with yellow. So purple goes with yellow because crossover has happened. Same goes on with the mother during the process of meiosis. If there is no crossing over, you just get parental kind of chromosomes, this and this. But if there is crossing over, then this fragment goes with green and purple goes with yellow. So that is what you get after crossing over. So these are all chance events. There can be crossing over, there cannot be crossing over. So it's all random. It all depends on the chance of whether crossing over has happened or not, and whether you end up getting a dominant allele or a recessive allele from parents. Now let's make a cross. If we make crosses like this, it will be so much harder and complicated because now we are dealing with 4 gametes on both the sides. Which is why it's always a better idea to use a Punnett square, and it's much easier to look through the Punnett square. Alright, so we have gametes from this filial 1 generation and the filial 2 generation. We are now going to build the filial 2 generation, and for that we will look at the Punnett square. Okay, now let's see what we get in the generation when we cross these 4 gametes together. So this is what we got from directly from the parent type, and these are the gametes we got after crossover. Again, for mother, these were the parental type, and these are what we get after crossing over. So let's make the crosses. This comes with this goes with this, and this type with this type. So you have to make elaborate crosses, like you end up with 16 different combinations. But let's decide what will be the phenotype between capital P and small p. If you ever have P and P, you know this will be purple. And if you have small p, you know this will be white. But when you have one capital P, one small p, it will again be purple because purple is dominant over white. Same with G's. Wherever you have capital G, this is green, easy to interpret. If you have small g, this will be yellow, easier to interpret. But if you have capital G and small g, what will be the phenotype? It will be green because green is dominant over yellow. Alright, so here you have capital P. This will be purple, green, purple, green, purple, green. So you don't have to think a lot. Wherever you see a capital P, it'll be purple. Wherever you see capital G, it'll be green. So purple, green, purple, green. So 1 purple, green, 2 purple, green, 3, 4, 5, 6, 7, 8, 9. So 9 purple, green. Let's look at purple, yellow. So capital P and both small g's. Capital P and both small g's. 1, 2, so 3. Now both small p's and 1 capital G. Both small p's and 1 capital G. 1, 2, 3. So 3 here. And both small p and small g 1. So there is 9 times chance of getting purple and green because they are dominant characters, and there's only 1 time chance that you will get both recessive characters, that is white flower color and yellow pod color, and there are equal chances of getting the mixed characters. Now we just saw the phenotypic ratio, how many greens, purples, whites, and yellows. Now let's also quickly look at the genotypic ratios. It'll be a little hard to see because there are so many letters going on, but let's try to follow a trend and we should be able to tell how many capital P, capital P, capital G, or a capital P, small p, both capital G. There'll be like 1, 2, 3, 4, 5, 6, 7, 8, 9 different combinations of capitals and small that you have. And if you want to count them, we will go kind of diagonally. So capital P, both capital P's and both capital G's, 1. This one is 1 and 2. This one is 1 and 2. So if you go diagonally, it's easier to count. This one is 1. This one are 4. 1, 2, 3, 4. This one is 1 time. This one is 2 times. This one is again 2 times. This one is 1 time. So you see that if you keep going diagonally, it's easier to count. So this is your genotypic ratio and this was your phenotypic ratio. Generally, phenotypic ratios are more important because the end result that you see in the offspring is what your answer is. That's what you're trying to investigate. So here we have concluded the dihybrid cross. In the next lecture, we'll study about the non-Mendelian inheritance. That means here it was very clear that one character is dominant over the other character. But sometimes there is no complete dominance, or it's not very easy to tell which character will be seen in the offsprings. So that is a non-Mendelian inheritance, and that we'll study in the next lecture.